So the first step is that we're gonna be creating the master mix in these 0.6 milliliter tubes. So like before, you're gonna wanna shake these out as opposed to reaching in. What we're gonna do is, for the ones that we're using, we label the top MM for master mix. If we're doing more than one type of PCR at the same time, then you're labeling specifically what type of master mix it is. So in our case, since you have one and I have one, I'm gonna label this as I, and yours is gonna be labeled as MMK. So the first step is to do the distilled water. Generally, I just go down by the order that's on here once you have the calculations. For the distilled water, it often has a point something after that. So when you're using the P200s, if you can see, there's these little tallies on there. Each of these tallies on the P200 stands for 0.2. So if you're at 71, in this case, since we're looking for 71.6, we go 1, 2, 3, and now we're at 71.6. So with that, when we're doing the water, we use double distilled water, which is right here. You're going to take it up. With PCR, it's really important to be exact. So you're gonna wanna watch as the water goes in. Pull it up very slowly, and afterwards check visually to see if it looks about right. This does look about right to me before putting it in. And again, you're gonna wanna watch it go in to make sure that there's the right amount in there. Okay. So the next step, as you can see, is to take up the 10x buffer. In this case, we're doing 10 microliters. You always wanna make sure that it's thawed completely before you use it, so there's no ice in here, as you can see. And then before you actually pull it up fully, you're gonna put the pipette tip in and then pull up and down a couple of times to mix it. And then sometimes when you do that, the pipette takes in a little bit less each time. So what I do is I push out fully, so there's nothing in the pipette tip, pull it out, and then go back in to pull up the final 10. And then you're gonna wanna, again, look visually to make sure it looks about right. In this case, we have the lines that show me that it's exactly 10. And then you take it in here. You put it in, and then when you put it in, you're again gonna push in and out a couple of times to mix it before pushing all the way out and lifting out. So again, going down the line, next step is the DNTPs. In this case, we're only taking up 2 microliters. So again, it's set to 2 exactly. And it's gonna be the same process as before. You're going to put it in there, pump up and down a couple of times just to mix it before pushing all the way out, lifting it up, and then going in and actually taking up the DNTPs. Looking to make sure it looks right. It looks about right to me. And then putting it in here, again, going in, pushing in and out a couple of times, for pushing all the way out and lifting up. Next up is adding the DMSO, dimethyl sulfoxide. Again, it's just the same process as before. In our case, we're doing 4 microliters, so just the same thing as before. Again, this looks like about 4, and so you put it in there. The dimethyl sulfoxide, when you're thawing it freezes really easily, so you really want to make sure that it is fully thawed before you use it. Next up, I do the primers. For the primers, it's especially important you make sure you have both the forward and the reverse primer, so that way you can isolate the gene of interest. So I do the forward primer first, which in our case is 1907 since we're doing Cas9. The same process as before. And then you want to make sure that you put in both primers and you don't accidentally put in one of them twice because they're very easy to confuse. So that's why I like thawing the primers individually, so I can keep track of which one I've already put in and which one I haven't. So this was the forward primer, and then next we're going to be doing the reverse primer. The next stage is the reverse primer, which in our case is 1908. Again, you want to make sure that it's the right primer before you use it because the numbers are very similar. Same process as all the other times. Pump in and out, push out entirely, and then pull up. Make sure it looks about right before putting it in, and then pumping up and down to mix it. Pushing it out entirely. You want to make sure that there's not liquid left in the tip, so you know that you've put it all in. Okay, so the last thing we add to the master mix is tack. With tack, it's very, very temperature sensitive, so it's important that when you take it out of the freezer, you keep it in this. It's the desktop cooler, which will keep it at the -20 that it has to be at. I only take it out of the freezer when I'm just about to use it as well, to ensure that it doesn't get too hot. For the tack, we usually end up using the P2 because it's a smaller amount, in our case 0.4, and P2 handles things that are 2 and below. So as you can see here, since these two are both red, this is the 1's place, that's the 10's place, so it's 0.4. For tack, it's especially important to have the exact right amount because it's very, very sensitive to that. So you really want to make sure that you look at exactly how much you're taking out. I usually hold it up to the window because it's easier to see. You're still going to mix it by pumping up and down a couple of times, push in, and then pull out, and then finally very slowly pull up the right amount. You want to make sure that it pulls it up fully. You can see that this looks like 0.4, and then you can put it in. Again, make sure that all of it goes in there. It's very important for tack especially. So the next step is to add the quat in your master mix, which is now finished, into your different PCR tubes. In this case, we have 3 PCR tubes each. So for our master mix, no matter how many tubes you have, you're always going to aliquot out 23 microliters of the master mix into each tube. Because of our calculations, we already know that we're going to have enough for all the tubes. And so again, you're going to mix it before pumping all the way out and then pulling up and putting it into the PCR tube. I tend to like doing the negative sample last when it comes to aliquoting, just in case there happens to not be enough. So because that way the one that has slightly less master mix is the one that's going to have the double distilled water as the negative sample. In this case, since it's the exact same thing going into clean PCR tubes, you don't have to use new tips in between samples because you're just aliquoting it. When you actually put in the DNA later though, you will have to use a new tip in between each sample. Our last step is just to do 2 microliters of each of the DNA samples for our samples that we're doing. So first, all of the ones that we're testing. Again, make sure the DNA is fully de-thawed. There's no ice in there. Push up and down a little bit to mix, and then go back in and pull all the way up to make sure it's about 2. And put that in the sample that you're looking at. You want to use different pipette tips in between each one so there's no worry of cross-contamination. Once you've done all the samples, in our case we're just having 1 each, you're then going to do your positive and negative control. So the positive control is a plant that you are certain will show positive for the gene of interest. In our case, we're testing for Cas9, and I've already tested this plant and it is positive for Cas9. So we know that if we did the master mixes correctly, it should show up in the Cas9. So you're gonna put that in your tube that's marked for the positive control. And then lastly, again using a new pipette tip, you're gonna do your negative control. Negative control is what will tell you to make sure that you haven't accidentally cross-contaminated anything. Your negative control is always just gonna be double distilled water, because that will not mix with anything in the thermocycler. And so if you did all the pipetting right and there wasn't any cross-contamination, it should always show up with no band. The next step is to make sure that your samples are all spun down fully into the bottom of the tube, because that'll make sure that the machine does it, like is able to actually thermocycle everything. So you're gonna put it in here in a centrifuge. I always hold my hand right above it because I've had samples fly out before, and then when they fly out you don't get them back, and then you're just gonna push this down and hold it for a few seconds in order to make sure that it's all spun down to the end. Then go into thermocycler. You're going to lift it up after you loosen it. You put them in. I tend to put them in towards the middle of it, just making sure that they're all all the way down in the machine before putting the lid back down. What you're gonna do is, you have it lined up right now to open. You're gonna spin it until you feel resistance and the red lines back up again on the other side, and you know now that it's quite tight. And then when it comes to the actual PCR machine, the first thing you're gonna do is you're gonna make sure that the settings are right for what you're doing. So for Cas9 at least, we use the one that's called GoTac-GE. You're gonna, after you go to edit, press proceed, go down to here, press proceed again, and then just check to make sure that it's all right. So it should be 95 for 2 minutes, 95 for 30 seconds, 60 for 30 seconds, 72 for 1 minute, multiplied 35 times, and then 72 for 5 minutes, and 4 forever, and then end. So that looks good, so that's fine. We're gonna go back to run, press proceed, main. Since you already looked at it for edit, it should be there as the first one, but either way you just want to make sure you're running GoTac-GE. This right now is in the B block, so you're going to press block, so it's switched to B, proceed, tubes, it's 25 microliters that are in each tube, use heated lid, and then now it's going to be running for about 2 hours. So the first thing when it comes to a lithium bromide station, before you do anything else, is actually cleanliness and safety. So first of all, lithium bromide is a potent mutagen. So this right here is the lithium bromide. Whenever you open it, you want to open it facing away from you, and you also want to close it facing away from you as well, because if it faces towards you, it could get onto your skin, and you don't want that to happen. You always wear gloves in the lithium bromide station. You do not use these gloves in any other station, and when you take them gloves off, you always want to do it without touching the gloves with your skin. You don't want to touch anything that's in the station with your skin, or any of the other areas that are lithium bromide contaminated, which includes this light switch, this microwave, this sink and back area, and if you come over here, this scale. All of these things are marked with this red tape which indicates that they have ethylene bromide contamination. So again, don't touch any of that without gloves on. Only touch it with your gloves, and don't use these gloves in any other location to avoid the spread of ethylene bromide contamination. When in the ethylene bromide station, you have two types of TBE. You have the 1x TBE, and in the back you have the 10x TBE. To make the gel, we use the 1x TBE. If you need to make more of it, it's a 1:10 ratio. So you would refill the 1x TBE using the 10x. When it comes to keeping the station clean, you always want to make sure when you're done using your samples, you put them, you toss them away unless you need to reuse them in the gel. When it comes to this, the rig, you always want to have the lid on there to keep the TBE from evaporating. Before you even make the gel, you want to refill the buffer to the fill line. Using the 1x TBE. And then what you're going to do is you're going to use our contaminated micropipettes in order to fill both sides of the rig with 3 microliters of the ethylene bromide. So you're going to use a tip. You can reach in to take these tips, but be very careful to only touch the end of a tip that attaches to the micropipette. You don't want to touch any of the actual tip ends, and don't reach fully into the bag. Just take whichever one is closest to the surface. Again, when it comes to the ethylene bromide, open it facing away from you, so that way it doesn't spurt onto you. You can take the 3 microliters and then put it into one side, mix it around. Next 3, other side, mix it around. And then, because whenever you run the rig, the ethylene bromide always migrates from this end to that end, what you're going to do, just to make sure it's all even, is you're going to pick this up and just move it back and forth gently, just to make sure that it's all mixed evenly throughout the entire rig. And that's the preparation. Cool. So to actually make the gel, the first thing we do is we set up the gel rig. We have the tray, which goes on the platform. You place it in, and then we want to make sure there's a really nice seal. So we first take this. There are these 2 holes here. You want to make sure that they aren't in there. So the holes have to be outside, so that way the gel doesn't go into it. And then we're going to take this. We're going to slide it in. It's going to be a bit tricky, but this is important to making sure that there's a very tight seal, so that way it doesn't slip out at all. Push it in there, and now this is very sealed. Next step is to make the type of gel that we use. We take 40 milliliters of the TBE and 0.32 grams of the agarose gel, which we'll do in a moment. So, first, the 40 milliliters of TBE. I'm going to pour it into here. It doesn't have to be exact, but it needs to be about 40 milliliters. As you can see, this is about 40. If you do extra, you can pour the extra into this rig. Don't pour it back into the TBE. And we put that there. Next, we're going to take the agarose gel. Using this, here's the tray we use. Go over to here. And we're going to measure out 0.32 grams. We want to do it sort of slowly to make sure that we don't go over. Again, it's okay if it's a little bit over 0.32, like 0.33 is fine, but we want to get it as close to 0.32 as possible in order to make sure that we have the right proportions for the gel. And then once we have that, we go over here, we take our flask, and I like to put this in first because if the flask is still dry, then there's less of a chance of the powder getting stuck on the sides. So I first put the powder in, and then I tilt it sideways and I pour in the buffer. The buffer will come out very quickly, so you don't want to pour like directly vertically. You always want to pour from a diagonal. And once you have this, you're going to take it to the microwave. Always use this microwave, don't use that one, because this one is contaminated and that one isn't. You put it in here, and then you're going to set it for 15-second intervals until it's boiling. You know it's boiling when even after you open the door, it's very bubbly. For me, typically, it takes 2 bits of 15 seconds and then 1 of 10 seconds. In between each interval, you're going to pick it up using the oven mitts and switch it around a bit before setting the next interval. You see here, it's bubbling a little bit, but this isn't boiling, so you still want to put it in for longer because you want it to be boiling. And now, as you can see, it is properly boiling because there's still production of bubbles even after I move it from the microwave. And so then, after that, you take it over here. You're going to put in 1 microliter of ethidium bromide into the solution. This is a P20, so normally it isn't supposed to go under 2 microliters. However, for the sake of what we're doing, since we don't want to contaminate a P2, we use the P20 even for things below 2 microliters. Again, open the ethidium bromide facing away from you. Take up carefully. Put it into here. Use the oven mitt to swirl it around to mix it. You want to make sure you swirl it rather vigorously because it's still fairly hot. So you want to make sure that the ethidium bromide really properly mixes with it. And then you gently pour it into the rig that you set up. You want to put this facing back down so that way the gel doesn't harden up on the bottom of this. And then the last step before you wait for it to harden is taking this ladder. You're gonna use it first to kind of like get rid of any bubbles that you see within the solution because you don't want bubbles in your gel. And then lastly, you're going to take the thicker end. As you can see, these are 2 different thicknesses. You're going to take the thicker end because that holds more DNA and put it on the far end of the rig. And that's going to sit for half an hour to harden before we can use it. So once the gel is ready to go, you need to prepare samples in order to actually go into the gel to run. So all the things we're going to be using to prepare the samples are in this brown fridge right here. Again, this is ethidium bromide contaminated, so you only touch it with your gloves and do not use any non-contaminated gloves to touch it. What you need is what's called 6X loading die, and you also need a ladder. I'm going to take these over here. where your samples are waiting. And the first thing you're going to do is to each of these samples, you're going to take a 6x loading die, not the ladder. They're the same color, so it's easy to confuse, but you want to make sure you're using the loading die. And you're going to add 1/6 of the amount of loading die of the microliters you have in here. So for example, we have 25 microliters in each of these tubes, so we're going to add 4.16 about microliters of the loading die into each of these. And you want to use different tips each time to avoid cross-contamination. I'm going to pick up the loading die. Take it, and then you're going to pump up and down to mix until the solution is solidly blue throughout. And you're going to do that for each of your samples. Okay, so now that we have all of these prepared, the next thing we're going to do is we're going to take our gel. It's been half an hour, so the gel should be firm and set. We're going to remove this comb by pulling straight up. Then we're going to carefully pull these out. Careful not to mess with the gel. And then we're going to take this entire tray and place it into here. It's going to go in between these like two parts. You see it sort of slides around a bit. That's okay as long as it's in between these two part areas. And then what we're going to do is we're going to take 15 microliters of each of the samples and insert them into the slots. When you're putting them into the slots, you want to hold the pipette above the slot. Don't insert it into the slot, because if you do, we might puncture it. So we want to put it above the slot instead. The very first slot is always going to be the ladder. You need the ladder in order to be able to compare the sizes of the bands that you might have. So you take the 15 microliters of the ladder, again, when you're inserting it, don't put it into the slot. Hold the tip right above the slot, stabilize with your hand, and then gently release so that it falls into the slot. And then you're going to do that with each of your samples, usually in the order of the samples you're testing and in the positive and in the negative. But as long as you have it noted down what the order is, you can do it in an order that makes sense to you. This is a little bit late because we already did the gel earlier, but after you have the gel in there, you're going to take this lid, you're going to slide it on in between these two slats here so that the black and the red line up with these little holes. And then when you turn on the machine, you go on. We use volts once it's like is turned on and ready to go. Select, make sure it's at volts, and then we are going to go up to 130. It can be a bit finicky sometimes, so sometimes you have to like try a little bit to get it to exactly 130. And then you're going to press run. We don't have anything in there right now actually, so I'm not pressing run. But you're going to press run, and then you're going to set a timer for 30 minutes. It runs for 130 volts for 30 minutes, and then after that you get to do the analysis. So after 30 minutes is over, what you're going to do is you're going to turn off the machine. You're going to slide this out, and then you're going to pick up this entire thing, sort of steady it with 2 fingers at the end so you can slide off the liquid without the gel sliding off. And then you're going to go onto here and you're going to slide the gel onto the UV like that. After that point, just like sort of tap out any bubbles. And now what's gonna happen is you're gonna have a hard time seeing this, but in order to see it, we have to have the UV on and we have to have these lights off. And now we can see. We usually take a picture of this. We want to like make sure it's really clear. So you see how it's already like fogging up. What you do is you sort of just turn it off. You shift this to a different position. And you turn it on and you get a picture real quick while it's still clear. So when we dispose of this, what we do is we take the gel, we put it in this container here. We turn the light back on so you can see. And then to clean this off, we're gonna spray both of these with the water, and then just wipe it down, both sides, leave it open to dry. And lastly, just for maintenance, I'm gonna put this back on so that way the buffer doesn't evaporate out. Take your samples. If you're not gonna run another gel, put them back here.